Hello everyone and welcome back. Today we're doing the specimen paper for 2025, theme B for higher level. Remember, this is the only paper that IB has released thus far, so make sure to pay good attention to this one. Let's get started. Starting off with paper 1A. So this question was also in the standard level. The diagram shows the elements present in two organic molecules, W and X. Which molecules could W and X be? Just to expedite this, I'm going to count down to 3. You can pause there, think about the answer. I don't want to rush you, but just so that this video isn't 2 hours long, that's how we're going to do it for every single question. So just pause now, and in 3, 2, and 1, it's C. Why? Okay, well, so if we look at A, monosaccharides do not have phosphorus, right? So that's already out of the picture. Then B, nucleic acids do have phosphorus, but triglycerides do not have nitrogen. So that's, again, not correct. And then D, triglycerides do not have phosphorus, and fatty acids do not have nitrogen either, making C the answer. Moving on, so what molecules are produced during the hydrolysis of a triglyceride molecule? This was also in the standard level. So 3, 2, and 1. Fatty acids and glycerol. So if you remember, a triglyceride is a glycerol molecule attached to 3 fatty acids, and that's joined via a condensation reaction. You can review that topic video if you don't remember, but the rest are incorrect because they don't mention these two ones together. Great. This one's long. So the structure of monomers affects the structure and function of the polymers they form. Which row describes the structural features of polysaccharides made from alpha glucose and beta glucose? All right, this might take a bit longer to think about. So 3, 2, and 1, and it's B. Why? Okay, well, you should know that starch is made of alpha glucose, whereas cellulose is made of beta glucose. Remember, cellulose is the only polysaccharide made of beta glucose, whereas starch and glycogen are made from alpha glucose. And then, so now you would be between A and B. And the reason why it's B is because starch is branched and cellulose is unbranched. Remember, that's really important because cellulose needs to be straight so that it can pile up and form those really, really rigid structures in plants. Good. So moving on, the diagram shows the structure of insulin over here. From the diagram, what can be concluded about the structure of insulin? All right, 3, 2, and 1. Okay, so this actually is taken from the 2024 paper. It's a very, very similar question, and this is a great piece of advice I have for you, okay? IB loves to reuse past paper questions. Even though this is a new syllabus, you can see that. So this question, if you go back to 2024, there's almost the exact same question. The diagram's a bit different, but it's basically the same question. So even though it's a new syllabus, you'll probably see a lot of the same questions. So the way to prepare for IB Bio, which people always ask me about, is to do past papers. Do more than you think you need to do. 5 to 10 years of past papers and you'll be set. But anyways, I digress. So you can see from here, it is composed of 2 polypeptide chains. So these are 2 separate polypeptide chains, and those are stabilized by disulfide bonds, these S's that you can see here, right? So B is false because you can see it's more than 1 polypeptide chain. It being a fibrous protein cannot be concluded from this, right? For that, we would have to see how it folds. And about not displaying quaternary structure, that's false because as soon as you have more than 1 polypeptide, you have a quaternary structure. Great. So, which is a feature of phloem sieve tube cells? Okay, this was also in the standard level. And 3, 2, and 1. They don't have a nucleus. So remember, the lignified walls are in the xylem, the chloroplasts are in the palisade mesophyll in the leaves, not in the stem. And it's not that they don't have a cytoplasm at all. They have a reduced cytoplasm, but what they don't have is a nucleus to allow for the pass of phloem. Next question. So in multicellular animals, embryonic stem cells have the ability to differentiate into a range of cells with different functions. What is the term used to describe cells with this property? And this is a bit of a trick question. So again, 3, 2, and 1. It's pluripotent. Why? Okay, I think a lot of you might have guessed totipotent. However, embryonic stem cells can differentiate into almost any cell type in the body, but they cannot form extra embryonic structures like the placenta or the amniotic sac. So they're not completely totipotent, right? They're more potent than cells like the ones in the bone marrow when you're an adult, but they're still not totipotent. Okay. So again, another one which was an SL. So the image shows a glycoprotein embedded in the phospholipid bilayer of a cell membrane. Which features of this glycoprotein can be deduced from the image? Okay, 3, 2, and 1. All right, so you know it's an integral protein. You can see it's embedded in the actual membrane, right? So it's not peripheral, so A is incorrect. And then Y is being correct because you can't really see it being a channel protein, right? You can't really tell that from the image. There doesn't seem to be a channel in there. And same thing with D. I mean, you don't, again, a transport protein that increases permeability to glucose, you can't tell that from the figure. However, it may be involved in cell recognition. We don't know for sure, but it may be, right? And that's something you can tell from the image. So always be cautious. IB is really, really annoying with the wording sometimes. So you have to be really careful. Okay, next question. So the diagram shows where the exchange of substances between blood and tissue fluid occurs in a capillary bed. What explains the movement of solutes between blood and tissue fluid at region X? So over here. Okay, and in 3, 2, and 1, it's C. Why? Okay, so you can see that blood flow is going in this direction, meaning region X is closer to the artery, right? It's the arterial end of the capillary bed. I know this question is kind of hard, but okay. So blood plasma has a higher concentration of solutes than tissue fluid. Now that's correct, but it doesn't explain the exchange at region X, because actually, in fact, blood plasma having a higher concentration of solids than tissue fluid means fluid from the tissue goes into the capillaries, but that happens at the venous end, so on the other side. Then B is incorrect. It's actually the opposite, right? And then C, that's true. So hydrostatic pressure is higher in blood than in tissue fluid. That's true. That's why at region X, because it's coming from the artery at a high pressure, the fluid is going to leak into the tissue from the blood. And then D, the permeability of capillary walls is highest at region X. Well, the thing is, the exchange of solutes between blood and tissue fluid happens because of pressure, not so much because of permeability. Like, it is, the capillary walls are permeable, but that doesn't explain why it moves in a certain direction in region X. I hope that makes sense. I can clarify in the comments if anyone needs more help in this regard. Next question, which was also an SL. So the number of hummingbird species in South America has increased continuously, as shown in the graph. What could be the reason for the increase in the number of hummingbird species during the period labeled set? Emphasis on species, okay? 3, 2, and 1. C. Why? Okay. Well, because, again, we're talking about hummingbird species, okay? So, A, if food and shelter are plentiful, and there's no competition, there's very little reason for more species to exist, right? So for more species to develop, you would just get a higher population of the species that already exists. Then B, closely related species can coexist indefinitely in the same niche. If that's true, again, same thing. C, okay, this is where it makes sense. So because if there are unoccupied potential niches, right, that means that competition within the occupied niches will lead to the creation of new species which can thrive and survive in these unoccupied potential niches, right? Leading to more species to develop. So hopefully that makes sense. Again, make sure to review the video on this topic if you don't remember. And then there are a few limiting factors on the population size of hummingbirds. Well, once again, then you would just get a higher population, not necessarily more species. All right. So, Yersinia pestis is a bacterium that caused an outbreak of bubonic plague in the 14th century. It normally produces ATP in the presence of oxygen, but can still produce ATP if oxygen is absent. Which term describes this characteristic? 3, 2, and 1. Pretty straightforward, right? So facultative anaerobe is what's called to organisms that can breathe with oxygen and without oxygen, right? If they're obligate anaerobes, they cannot survive in oxygen, and if they're obligate aerobes, then they require oxygen to survive, and then A is just not a thing. Next question. What are common features of holocellular nutrition and saprotrophic nutrition? Again, question from SL. 3, 2, and 1. Okay, so whilst it's true that saprotrophs are mostly fungi and bacteria, that's not true of holocellular nutrition, so they don't have that in common. So, however, both secrete enzymes to digest food. Just remember that in holocellular nutrition, it's internal digestion. In humans, for example, in the stomach, we digest the food. Saprotrophs release the enzymes, but it's still secretion, nevertheless. And finally, ingestion of food particles. That's only true in holocellular nutrition, internal digestion. In saprotrophic nutrition, remember, they just ingest the nutrients after digestion. Okay. Next question, again from SL. So the preferred temperature ranges for 3 species of trout that are found together in freshwater lakes in North America are shown. What can be deduced from this information? In 3, 2, and 1. 1 and 3. Why? Okay, so it's clear that 1 is correct, right? The niches all overlap. I mean, you can see that the temperature ranges overlap in all of them. 2 is false because brown trout over here do not have the most specialized niche, right? I mean, they share a big part of the range with the rainbow trout. Actually, the lake trout, if any, is the most specialized. And then lake trout can avoid competition by living in cold water. That's true, right? They have a range from 6 to 11 where none of the other trouts can live. So they avoid competition in those ranges. So that's why 1 and 3 are correct. Amazing. So that's the end of paper 1 and, well, 1A. And now we're moving on to paper 1B. Okay, so this one's really long. Beetroot tissue was cut into pieces of similar size and shape. The masses of the individual pieces were measured. 3 pieces of beetroot were placed in each of the 6 sucrose solutions of different concentrations. You can see the concentrations here. After 3 hours, the pieces were removed from the solutions, surface dried, and their masses recorded. The mean percentage change in mass of the beetroot in each solution was calculated and displayed in the graph, which you can see over here. All right, so the first question is state the independent variable in this experiment. This should be easy. 3, 2, and 1. It's the sucrose concentration, right? That's the independent variable, and the dependent variable is the change in mass. Next question. Outline the reason that beetroot pieces of similar size and shape are needed in order to obtain reliable results. Okay, so this is again about experimental design. 3, 2, and 1. Okay, well, because if you have them being different sizes and shapes, they're going to have different surface areas, and that affects osmosis, because remember, osmosis happens across membranes, right? And that is determined by the surface area. All right, next question. Suggest a reason for expressing the changes in mass as percentages. In 3, 2, and 1. To have fair comparison, right? So you actually might have pieces which had an initial different mass, even though you're trying to make them the same shape and size. That's impossible to do perfectly, right? So you do it as a percentage to make sure that it's a relative measure, right? And it's not in absolute terms, because that would be unfair to compare. All right. So then explain the effect of the 0.0 mole per cubed decimeter sucrose solution on the water potential of the beetroot pieces. All right. So again, 3, 2, and 1. Okay, so at a sucrose concentration of 0, meaning it's water, right? Water is going to enter the beetroot. Why? Because the beetroot is going to have a higher solute concentration. So that's going to happen via osmosis, meaning pressure potential is going to rise, increasing water potential of the tissue of beetroot. Eventually, an equilibrium will be reached when the cells are turgid. Remember, that's when they're isotonic, right? So you have the same concentration of solute inside than outside. So hopefully that makes sense. I know I'm going quite quickly through these. I think they're semi-easy, but if you have questions, again, leave them in the comments. So that's all for paper 1B for theme B. So now moving on to paper 2. All right, so I know this is super long, but we're going to read through it slowly. So squirrels have evolved several defenses against predators. One method adopted by California ground squirrels against rattlesnakes is to chew molted rattlesnake skins, then lick their own fur so they take on the scent of a rattlesnake. Okay. An experiment tested the amount of time rattlesnakes spent studying and flicking their tongues at filter paper soaked in different liquids. The snakes were released from chamber C into foraging area F and observed for 40 minutes. There were 2 filter papers in area F. One was a control paper soaked in distilled water. The other was soaked in 3 types of scent: squirrel scent, a mixture of squirrel and rattlesnake scent, and rattlesnake scent. Okay, so you can see up here in the diagram, okay, so the snakes are released from here, from chamber C, and then they're released into this foraging area, this big square, and the filter paper, the 2 ones are here, right, a control, and then one that's soaked in different scents, and the position is changed at each trial. Okay, so hopefully that's simple enough. And then this is the time studying the filter paper. And then this is the number of tongue flicks. Okay, so first, calculate the difference in mean time spent studying squirrel-scented filter paper with and without rattlesnake scent. 3, 2, and 1. about 80 seconds, right? So if you look at this graph and you look at squirrel scented paper, about 160 seconds, and then you look at the rattlesnake scent plus squirrel, right? It's about 80. The difference is about 80 seconds. Okay. So the next question is outline the conclusions that can be drawn from the control results. In 3, 2, and 1. And it's that snakes spend a very short amount of time studying the controls. So you can see the controls here, right, in gray, and it's very, very short in all three compared to the real scents. And that's a good thing, because that means that the filter paper itself is not a factor in the experimental results. So the filter paper is just a way to do the experiment, right? We actually don't care about the filter paper itself. And actually, ideally, we would have squirrels, right? But for ethical reasons, I imagine they used filter paper. So we want to see, we want to make sure that it's not that the filter paper itself is attracting the snakes. Hopefully that makes sense. Okay. So the next question is the positions of the filter papers were changed for every trial. Outline one reason why this was necessary. In 3, 2, and 1. Okay, so obvious, right? The snakes could just learn where the filter papers are and go there instead of according to scent, which would throw off the entire results of the experiment. Good. The next question is suggest a reason for rattlesnake scent lowering the chances of snake attacks on squirrels. In 3, 2, and 1. Okay, so it either masks the scent of the squirrel, meaning they actually can't smell the squirrel, or it could also suggest the presence of a rival snake, right? And then they don't want to get in there to avoid conflict. Next question. So now we have a new graph, right? So now the researchers observed that when squirrels saw a rattlesnake, they waved their tail from side to side so that they would be more visible to the rattlesnakes, and that's called tail flagging. The graph shows the probability of a snake strike at different distances on tail flagging and non-tail flagging squirrels. All right. And now the first question is compare and contrast the effect of tail flagging with non-tail flagging on strike probability. Okay. And so tail flagging is the triangles and non-tail flagging are these rhomboids. So 3, 2, and 1. Okay. So obviously at shorter distances, right? So this is the distance between the snake and the squirrel. At shorter distances, they both have a high probability of a strike. It doesn't really matter, right, whether they tail flag or not. But at greater distances, you can see that they have a low chance of strike if you tail flag, but it's still a high chance if you don't tail flag. That's very, very clear, I think. Okay, so for the next question, estimate the distance between squirrel and snake where there's a 50% chance of a strike at a tail flagging squirrel. Okay, so this requires you to take out some pen and paper. So pause now, and in 3, 2, and 1. Okay, so about 14 centimeters. So how do you actually calculate that? Okay, so if you're looking at the triangles, the tail flagging, right? This is 100% and this is 0%, right? So if you draw a line between here and here, right? And then on that line, you find the point at which you have a 0.5 probability. So it'll be around here. That's about 14 centimeters. 15, 13, 16, probably fine, right? But as long as you show you're working and give a somewhat reasonable value, you should be good. So next question. Suggest a reason for the behavior of snakes towards tail flagging squirrels. 3, 2, and 1. Okay, so if they're flagging their tails, that means the squirrel is probably alert and knows what's going on, so the strike is more likely to miss, and that would be wasted energy for the snakes, so they would rather not strike. Okay, so the next question has yet another graph. We can't get enough of these squirrels. So when played a recording of the call of a rat-tailed hawk, foraging gray squirrels in the wild became more vigilant and looked up from their feeding more often. In the habitat of squirrels, there are also sounds made by songbirds, which chatter to each other when there's no imminent threat. The charts show the response of the squirrels in 30 seconds immediately after the scare of hearing hawk sounds and after 3 minutes, both with and without bird chatter playing. Okay, that's a lot, and actually I feel like, at least for me, that takes a while to process, so maybe read it again. But the question is, state the conditions that led to the squirrels making the least mean number of lookups. So again, pause now, take a minute or so, and in 3, 2, and 1. Okay, so if we look at the number of lookups over here, right, that's what we're looking at, and we're looking at the least mean number that's clearly here, right? It's with chatter and 3 minutes after, so the white, right, not the stripes. The next is discuss briefly whether the sound of a bird chatter represents a significant difference in the vigilance of squirrels immediately after they have heard the sound of a hawk. In 3, 2, and 1. Okay, so again, we're looking at vigilance here, maybe, right? So the second graph, percentage of time spent vigilant, and we're also looking at immediately after, so the striped lines, right? And you can't see a significant difference. I mean, there's a humongous overlap in the error bars, right? You would have to perform a statistical test, but it's very, very unlikely that it would yield a statistical significance. Next question is, using the data in the bar charts, analyze the effect of bird chatter on the behavior of squirrels. And you can say a lot of stuff here. So 3, 2, and 1. Okay, and you don't need to say all of this, but these are some ideas. So the data supports suggestion that squirrels listen to bird chatter, right? Clearly there's a difference in response between chatter and not chatter. Bird chatter is reassuring as it indicates no imminent threat to the squirrels. That might very much be true. With chatter, the squirrels were less vigilant. That's clearly true here, right? At least after 3 minutes. And the data only shows a significant difference in the response with chatter. That's also true, right? You can see that clearly here. However, large error bars means conclusions may be unreliable. Again, that's true, right? In most of these, the error bars are huge, right? Which is not very good when you're analyzing data. Okay. And finally, we can move on from the squirrels. So only a couple questions left. So multicellular organisms need efficient exchange surfaces where molecules can move between tissues and their surroundings. In mammals, gas exchange takes place in the alveoli of the lungs. Describe how the exchange of gases is brought about between the alveoli and the blood for 3 marks. And in 3, 2, and 1. Okay, so the concentration of oxygen in blood is lower than in air, right? Because of we use up the oxygen, right, for cellular respiration. Then oxygen moves into the blood by diffusion. Oxygen concentration in alveolar air is lower than in atmospheric air because it's moved into the blood, right? That makes ventilation occur, right? So replacing that oxygen into the alveolus, so the air is completely replaced. And the flow of blood in the capillaries maintains the concentration gradient. You can give this answer exactly the same, but switching it around and talking about CO2, right? Or you can do both, talk about both. But for 3 marks, you don't really need to, right? As long as you mention some of these aspects, you are good. And the last question. So describe 2 features common to the exchange surfaces of alveoli and gills. Okay, and you have some images here. So in 3, 2, and 1. Okay, so they both have a rich capillary supply, right, for gas exchange. Then the epithelium is very thin to allow the gases to actually permeate. Moist surface, that allows the gases to pass through as well. And a large surface area, right, to allow more gas to pass through these surfaces. Okay, so I know that was pretty long, and some of these are quite hard, but hopefully that was clear. Any questions, leave them in the comments. I've also noticed that a lot of you who watch aren't subscribed, so please do, because it really helps me out. And I'll see you in the next week. We'll start with theme C. Goodbye, everyone.